Good evening. This is Dr. Najeeb. I hope you are doing well, right? And I hope you must have watched some of the lectures which I have given already. So today we are going to talk about a very special topic, right? We are going to talk about how the blood glucose level is regulated in a very narrow range. In a previous lecture, we discussed that normally in a healthy person where blood glucose regulating mechanisms are working well, in a healthy person, blood glucose level should not below 50 to 60 milligram per dl or 100 ml. And on fasting level, fasting blood glucose should not be more than 100 milligram per dl. Listen carefully. Body regulates blood glucose level in a very, very narrow range, which is non-toxic range. Glucose level below that is also toxic and above that is also toxic. In both conditions, if blood glucose level is less than normal, it damages our body. If it is more than normal, it damages our body, right? So our minimum blood glucose should be maintained around 50 to 60 milligram, and fasting blood glucose level should not be more than 100 milligram, and 2 hours after the meal, we call it 2 hours postprandially. 2 hours postprandially, blood glucose should not be more than 140 milligrams per dl, right? And randomly, our blood glucose should not go above 200 milligram per dl. Now, why I put it black here and black here? Because if blood glucose level goes above these values, right, that will produce damage in our body, as we discussed in our previous lectures, that high blood glucose chronically, that is hyperglycemia, that eventually leads to many complications, including macrovascular diseases, microvascular diseases, and infections, and many other complications. In the same way, in the previous lecture, I mentioned if blood glucose level falls too below, that produces symptoms of hypoglycemia. Even it can lead to the sympathetic overflow and then related symptomatology, and eventually lead to hypoglycemic shock. So what I'm trying to say, that our blood glucose level should remain in a normal range, in a very narrow range, and our biological system has very strong defenses against hyperglycemia and against hypoglycemia. Again, I will repeat it. Our biological mechanisms in our body, our organs and our hormone system, they interplay with each other in such a way that despite that our food intake and our physical activity is very variable. Our food intake, the amount of food, the contents of food, the timings of food are very variable. Our physical activity is very variable, right? Yes. But blood glucose is tightly controlled in a special range, right? How it is controlled that, right? The reason being we have special biological mechanisms today I will talk about, which do not allow the blood glucose to go above the normal range. That is called defenses against hyperglycemia. In the same way, our body has special mechanisms to prevent the blood glucose to fall too low, and they are, as a group, those mechanisms are called our defenses against hypoglycemia. Our defenses against hypoglycememia. Today we will talk about that when you have eaten a lot of food. If you have eaten a lot of food, let's suppose we take an example of a man 1000 years back, 1000 years back in a beautiful, very small village, there is a man who is a woodcutter. And in the morning, every morning, he goes to cut the woods in the jungle. Now, let's suppose we talk about some special morning, that in the morning he took very good food, he took lots of carbohydrates, some proteins and some fats also. He was full. It means he entered into his gastrointestinal system and his biological system of that man entered into well-fed state. In which state? Fed state. Fed state means that his gastrointestinal system, right, that is very rich in nutrients. Gastrointestinal system is very rich in nutrients. He has taken a very good breakfast and now he has gone to jungle. Now, what will happen? That when he has taken very good food, right, and he has gone to jungle, from the gastrointestinal system, there is absorption of nutrients going on, digestion and absorption, right? For example, gastrointestinal system is absorbing glucose, it is absorbing amino acids, it is absorbing fatty acids, and with that, like potassium present in the diet and phosphates and many other nutrients and vitamins, right? So we say that he is in a well-fed state, right? Now, this well-fed state, if you focus only on glucose, if lot of glucose enters into blood, right, because he has taken a very good meal and due to that reason, lot of glucose is absorbing from the gastrointestinal system. If this glucose goes directly to the systemic circulation and it is not processed properly, it will produce a very big spike and it will produce hyperglycemia, and that hyperglycemia is toxic for the body, as I mentioned in the previous lectures. So, what we will see for this man, while he is going and he is in fed state, what are the different biological mechanisms, what are different organs, what are different hormones, what are different metabolic pathways, what are the enzymes involved which are responsible to prevent the hyperglycemia. What I mean by prevention of the hyperglycemia, that in this person, after taking the meal, within 2 hours, his blood glucose should become less than 140, right? All of us know that to prevent the hyperglycemia, insulin plays a major role. So it means insulin and its related other activities on different organs that should, within 2 hours of the meal, should bring the blood glucose level less than 140 milligrams per dl. Let's see how the events unfold. We'll start from the gastrointestinal system, right? That from gastrointestinal system, let's suppose here is your GIT, okay, believe me, this is GIT, right? So gastrointestinal system, let's suppose it is very rich in nutrients, right? It is very rich in nutrients, and nutrients are being absorbed. In special circulation, we call it portal circulation, right? Now, here is our very lovely organ, everyone knows, this is pancreas, because pancreas plays a very big role. Pancreas has exocrine functions as well as endocrine functions, right? And its endocrine functions in the pancreas are limited to special type of collection of cells, and those collection of cells is called islets of Langerhans, right? Now, when this glucose is coming, amino acids is coming, yes, fatty acids are coming, right? They are passing through the circulation of pancreas, and then pancreatic circulation, and gastrointestinal circulation, right, and splenic circulation, all of them make a special vein. We call it portal vein. What we call it? Portal vein, which will take these nutrients and substances released by the pancreas together to our lovely organ, that is liver, right? Now what happens in pancreas? First of all, as nutrients are coming to here, now this is, you need to focus very well. As nutrients are coming over here, not only nutrients are coming, because within the mucosa of gastrointestinal system, there are special cells. There are special cells which act as nutrient sensors. Look, again I will repeat. In the lining of mucosa of GIT, right, especially in small gut, not only there are cells which absorb the nutrient, but there are special cells also present which are neuroendocrine derivative cells, special cells, and these special cells act as sensors of nutrients. They can sense the presence of glucose or amino acid, and what these cells are doing, these cells release special type of substance which is called as a group incretins, right? So let's make it here that gastrointestinal system is releasing, not only absorbing the food, but its neuroendocrine special type of cells, they are releasing special type of hormones. They are releasing special type of hormones, and these hormones are called incretins. As a group, they are called incretins. Now, these incretins, there are many types, but two of them are very, very important. One is GLP-1, and that is glucagon like peptide 1, and other is called GIP, that is glucose dependent insulinotropic peptide, which was previously called gastric inhibitory peptide. Now these two substances, they are called incretins, and they are released from the gastrointestinal cells. GLP is released from L cells, and GLP is released from L cells, from the small intestine, right? And GIP is released from the K cells, right? Now what is the purpose of these? Question is that what is the purpose of these incretins while person is moving in the jungle, stomach, his gastrointestinal system is full of nutrients, lot of nutrients are being absorbed, and along with the nutrients, small intestine is releasing incretins. Now what is the purpose of these incretins? These incretins, number one, these incretins are going to pancreas. They are going to pancreas, and especially on the beta cell, in islet of Langerhans, right? In the pancreas, if I draw an islet of Langerhans, what happens that in the center there are beta cells. In the one islet of Langerhans, in the center there are beta cells, and around that, beta for beautiful, they are beautiful cells which release insulin specially, and then around it the alpha cells like alpha males. They are on outer side, beautiful ladies are in the central group, alpha males are around that, and then there are some delta cells which are like dogs, delta cells, and dogs are everywhere. They are in the central part as well as outer part of islet of Langerhans. Let's suppose this is islet of Langerhans. What I'm trying to say, this is a small endocrine organ, right? And there are about 1 million islets present in, what is this? Pancreas, right? Central cells are beta cells. Around them there are alpha cells, and the delta cells spreading all around. Beta cell mainly produce insulin and other substances which I will tell later. Alpha cells mainly produce glucagon. At this stage, that is enough for us. Now, what these incretins are doing, what is the function of this incretins which are being released in this man? Let's suppose we call this man jungle man, fully loaded with the food. So in this jungle man, what is happening from the GIT, along with the absorption of the nutrients, GIT is also sending special chemical signals, and these are called incretins. Now what these incretins are doing? Number one, they are going to act on the beta cell. They are going to act on beta cell. On the beta cell, they are having special receptors, 7-pass receptors, and incretins stimulate the beta cells, and in the presence of incretin, now listen very carefully, normally beta cells are stimulated by what? Beta cells are stimulated by the arrival of glucose, which is a very strong stimulant to beta cell to release insulin. Then amino acids also relatively weak stimulant, fatty acids most weak stimulants, right? And then these three nutrients, and then there are incretins, right, which is coming from small gut cells, K and L cells, and then there is cholinergic stimulus. As you know that cholinergic stimulus is secretomotor to gastrointestinal system. So when you have eaten a lot, right, then parasympathetic system to the gastrointestinal system is firing and releasing lot of acetylcholine. So main stimulant for the beta cells are glucose, amino acids, and fatty acids, but most strongest among them is the glucose. Then those beta cells which are releasing insulin, under the influence of arrival of glucose, if they are also exposed to incretin, the release of glucose becomes amplified. It is very important. Again, when glucose reaches beta cell and when it stimulates the beta cells, then glucose, when it is releasing, if glucose is alone and no incretins, then less insulin is released. But if glucose is stimulating the beta cell, at the top what comes? Incretins, insulin release will become amplified. It means one very important function of incretins is glucose induced release of beta cell is amplified. It is increased. So it is good, number one, because more insulin, better control on the glucose. Number two, these incretins will act on the stomach. They will act on the stomach and reduce the motor activity of the stomach, so that while lot of food has already arrived, right, semi-digested food, we call it chyme. When chyme has reached in the small intestine, while small intestine is dealing with this food, it is digesting this food, it is absorbing this food, sending the nutrients to pancreas, and also sending the incretins, right? So at the same time, small intestine should send some signal to the stomach that slow down please, let me deal with whatever is coming, which has already arrived. Now the signal back to slow down the stomach is also incretins. So now we know two functions of incretins. Again, this jungle man has eaten a lot of food. He is in fed state and absorptive state and absorbing a lot of nutrients. Small intestine releasing from special cells which are neuroendocrine cells. From there it is releasing incretins. Incretins and nutrients have, right, incretins have many functions. Number one, incretins can go to pancreas, act on the beta cell and increase the release of insulin. Number two, incretins can go to stomach and slow down the stomach, that please relax, let me deal with whatever chyme or semi-digested food has already arrived, let me deal with that. Then slowly release more food to the small intestine. Thirdly, incretins act on your hypothalamus. incretins, right? This is hormone, right? Incretins which are released from the small intestine not only act on beta cell, not only act on the stomach, they also through the blood eventually reach to the hypothalamus, and on the hypothalamus they stimulate the satiety center. They stimulate the satiety center, so person knows that he is having lot of food already, and if someone present him some food, hopefully in jungle no one is presenting, but if somehow from somewhere someone comes and offers more food, he will say, no, I am already full. You are getting it? It means his satiety center is also firing, that he has eaten well. Now, here I want to, there is a very important clinical point related with incretin which I want to enhance, that let's suppose this is release of insulin levels, and here is the suppose time, right? When you stimulate the beta cell, right, insulin is released. Now, there are two ways to force the insulin to release. Number one, if you eat the food, it goes to small intestine and glucose will go and stimulate the beta cell, and incretins will also go and stimulate the beta cell. Another way is if you give the person intravenous glucose. If you give the person intravenous glucose, do you think small intestine will release incretins? No. If you are giving a person intravenous glucose, even though there is no intravenous system in jungle, but anyway in our jungle life it is there. So if you give intravenous glucose or exogenous glucose which is given in systemic circulation, right, does not stimulate the small intestine to release the incretins, but does stimulate the beta cell to release the insulin. So if you give exogenous, let's suppose glucose, then insulin level will go up, but it will have less peak and eventually it will come down, insulin level. Okay. Now, there is some basal level going on now. But if you take the food, what will happen? Your insulin level will go higher level. Insulin release, when you take the food, insulin release will be at higher amount and for little longer time. Why? Because insulin release is amplified here. This is insulin release when glucose is given intravenously. This is insulin release when carbohydrates or glucose is given orally. This is the insulin release when beta cells are activated by glucose but without effect of incretins. And this is amplified release of insulin, right, which is there when we take glucose orally or carbohydrates orally. In this case, beta cells are not only stimulated by glucose, but beta cells of pancreas are also stimulated by incretins, and due to incretin there is amplified response. This difference is called incretin effect. Incretin effect. Right? Why it is so important? Because these days we are having certain drugs, right, which are mimicking the structure like incretins, and they are used to amplify the release of insulin. Is that right? Now, let's go back. What we have learned up to now, lot of food eaten, small intestine is reabsorbing lot of food and nutrients, digesting and absorbing the nutrients. Along with that, in fed state, small intestine is releasing incretins. Incretins have three important functions. Number one, they amplify the release of insulin, and number two, they are reducing the motility of stomach, and number three, in the central nervous system, they are acting on hypothalamus and leading to satiety, feelings of satiety. Satiety center are stimulated, right? Now what happens? Let's move forward. Here was our, let me make the main organs where glucose handling will be done and utility of glucose will be done, right? First organ which I would love to talk, I have already talked, and that is about gastrointestinal system. Then we talked about the next organ, that is pancreas. Then we talked about that pancreatic blood flow and gastrointestinal blood flow and splenic blood flow. It all together is going through portal vein, and this portal vein is taking the blood, all these nutrients along with incretins, all along with the insulin, everything towards the liver. So liver will be the next big organ in this game. Liver will be the next big organ into this game. Then we will see later that while handling the glucose, what is the role of the heart, what is the role of the muscles, and what is the role of the skeletal muscle. Okay, he is laughing at you. And what is the role of adipose tissue? This is, let's suppose, adipose tissue, right? And in the end we'll talk about brain, which is not dependent on insulin for its uptake of glucose, right? And we'll talk about later the role of kidney, especially in fasting state, when there is some gluconeogenesis done here in fasting state, not in fed state. And in fasting state, we'll also talk about suprarenal gland, which releases cortisol, and from adrenal medulla, it releases epinephrine, catecholamines. Now, these are different organs. These organs have to interplay. We have only learned very basic, nutrients, beta cells. Now, let me go little detail on that how glucose work on the beta cell. I will bring a beta cell here, right? This is a little revision from previous lecture, right? So what is happening, that this is a beta cell from the pancreas, beta cell. From the pancreas, islet of Langerhans, right? Now, first of all, glucose has to enter into cell. Now, cell membranes are inherently not permeable to glucose. So there in the membranes there are special type of transporters which transport the glucose from extracellular site to intracellular site. Now, there are many types of different glucose transporters. There are many types of different glucose transporter. Here we have a very special type of glucose transporter, and this glucose transporting channel, right? This glucose transporting channel is called GLUT2. What is it called? GLUT2. Now, these GLUT2 are naturally present on the beta cells of the pancreas, right? These special type of glucose transporter, type 2, they have something special. They let the glucose move in any direction. They can allow the glucose to move in any direction depending upon the concentration gradient. If there is more glucose outside, glucose will move in. If glucose is more inside the cell, through the GLUT2, it will move out. So, what is very special thing about GLUT2? Number one, they are always present on the specific membranes, right? I will tell you what are those membranes, cell membranes. Number two, they are not insulin dependent. These transporters are not insulin dependent, right? Insulin is there or not there, they are open there, right? Number two, they let the glucose move in either direction, from extracellular fluid to inside or from inside to outside, depending upon glucose concentration gradient. Is that right? Now, GLUT2 are present, of course, where? Number one, of beta cells of pancreas. Number two, GLUT2 are present on the hepatocytes, liver cells, and then GLUT2 are also present on basolateral membrane of gastrointestinal cells, mucosal cells. Let's suppose this is a gastrointestinal cell, right? This is a cell, right? Now this is which side of the cell? Luminal side of the cell. What is this? luminal side of the cell, right? And this membrane of the cell is called basolateral membrane. What is this? Basolateral membrane. It means if I draw one luminal cell, this will be the basolateral membrane, basal and lateral, and this is luminal side. Now, these cells in small intestine and these cells, similar cell, absorptive cells in nephrons, for example in proximal convoluted tubule, because in GIT also we absorb the glucose, in proximal convoluted tubular nephrons we also absorb glucose. Nephrons also have luminal side and basolateral side. Now, on luminal side, there are special mechanism to take up the glucose in, right, into this cell. But on basolateral side, there are present GLUT2. What are these present? GLUT2. And when you have taken glucose-rich food, then glucose by special mechanisms transport mechanisms will be taken up from the lumen to the cells, and from the cell, glucose will escape basolateral side towards the extracellular fluid and then into circulation through the which type of glucose transporters? GLUT2. So GLUT2 are present where? On the beta cells, they are present on hepatocyte, they are present on lateral side of the, please not luminal side, basolateral side of absorptive cells of gastrointestinal system, GIT, and also absorptive cells in nephron, in kidney, nephron tube, in kidney, right? These are the four places where GLUT2 are present. Is that right? And the beauty of GLUT2 is that they let them move glucose in any direction. That is, 2 means two-way. For us to remember, GLUT2 means glucose can move in two-way. Now, as I mentioned in previous lecture, that when glucose will come from the gastrointestinal system in high concentration, right, big amount of glucose will start moving inside the, what is this? Beta cell. So inside the beta cell, glucose level will go up. So this jungle man, while he is moving in jungle, he is absorbing glucose. Glucose levels in the circulation around the beta cell become very high. Then this glucose will start entering into beta cell via the GLUT2. Now this glucose will enter. Inside there is an enzyme which is called, this enzyme, yes, this enzyme looks very serious. This is called glucokinase. What is the name of this enzyme? Glucokinase. Kinase means that this enzyme is going to add phosphate to the glucose. So this glucose will convert under the influence of glucokinase into, yes, glucose 6-phosphate. Because glucokinase will lead to phosphorylation of the glucose which is taken in, right? Now, once it is converted into glucose 6-phosphate, now listen, as from gastrointestinal system, more and more glucose is coming, more and more glucose is entering through the, which channel? GLUT2, inside the beta cell. Then this enzyme, glucokinase, they are activated, and these glucokinase are activated by basically presence of glucose, and they will convert glucose into glucose 6, and when they will convert glucose into glucose 6-phosphate, right, we say glucose is trapped, because now phosphate will bind with glucose and make the glucose phosphate complex very, very polar, and it becomes so heavy that it cannot move out. Secondly, when glucose is coming in, it keep on converting into glucose 6-phosphate, so intracellular glucose level remain low and extracellular glucose level remains high. So when this glucose convert into glucose 6-phosphate, eventually it is taken into glycolytic pathway. I will not go into detail of glycolysis, right? Eventually pyruvate is made, and this pyruvate will enter into Krebs cycle, right? It will enter into Krebs cycle, and from there energy will be taken by, what are those? Electron transporters, and the end result will be that in the cell ATP levels will rise, right? So it means more carbohydrate you eat, more glucose is absorbed, more glucose passes through GLUT2 to beta cells, more ATP is formed, more glycolysis in the beta cells, more ATP is formed. First glycolysis, then Krebs cycle, and then electron transport chain. Eventually ADP is converted into ATP. Now this ATP, everyone knows, every good doctor knows, ATP is a very good source of energy. But here I will talk about very unusual function of ATP. The ATP here molecule, let's suppose this is a molecule of ATP, right? This has a very, very special function. You know, it is laughing. Why it is laughing? It is going to activate a mechanism to release insulin. So what will happen, that this ATP molecule, it will act on special type of potassium channels. What are these? Potassium channels, and these potassium channels which are present in the membrane, they are sensitive to the presence of ATP. Actually, truly speaking, this ATP can go, molecule can go and yes, ATP. What it will do, it will basically plug this, it will close this channel. So what happens, that as time passes, more and more glucose is being absorbed and beta cells are producing more and more ATP, and more ATP is blocking more and more ATP sensitive potassium cells. Which cells? So potassium channels, potassium channels. So when ATP sensitive potassium channels are blocked, normally our cells keep on losing a slight amount of potassium. Very slight amount of potassium from most of our cells is all the time going out. And this loss of cations keep the interior of the cell, inner side of the membrane, due to loss of potassium, relatively polarized to the negative side, polarized towards the negatives. Due to normal loss of potassium, here normally it is minus 70 millivolt, right, voltage inside the membrane. But what happens, when more glucose comes, more ATP is generated, more potassium channels are closed, now potassium cannot go out, potassium cannot escape out. If potassium does not go out, it is retained inside, and when potassium is retained inside, it is kept inside. What will happen, that inside will become, potassium is positively charged. So when positive charges are retained inside, what will happen, that inside will become gradually more towards the positive side, or initially it will become less negative. Because potassium is retained, let's suppose it become minus not 70, right? Let's suppose this was minus 70. Gradually, due to retention of potassium, right, this resting membrane potential gradually moves towards positivity. Let's suppose this is 0. But when it reaches about minus 40 to 50 millivolt, minus 40 to 50 millivolts, what happens? Again, potassium channels are closed, and closure of the potassium channels lead to the slight retained potassium. This retained potassium in the cells makes the cell's interior less negative, and from minus 70 millivolt resting membrane potential, then due to retention of potassium, right, these cations will pull the resting membrane potential towards positive side, or make it initially less negative. When it reaches to minus 40, something special happens. What is that something special? That in the membrane there are also calcium channels which are voltage sensitive calcium channels. These are calcium channels, right, and these are called voltage sensitive calcium channels, or voltage regulated calcium channels, or voltage dependent calcium channels, right. Now, as soon as due to blockade of the potassium channels, potassium is retained in cell become less positive, less negative, and when voltage of the cell from minus 60 or minus, first it is minus 70, then minus 60, then minus 50, when it is more positive than minus 50, it become like minus 45 or minus 40, suddenly these calcium channels sense the presence of what? Change in the voltage, and these voltage dependent or voltage sensitive calcium channels will open, and when they will open, lot of calcium will start moving in, and this calcium which will come in, what this calcium will do? This calcium will work on the preformed granules. Preformed granules, these are the granules which are having, number one, they are having, let me make it, let's suppose this is a peptide chain, these are all amino acids, and I call it, it's a very long chain, we call it pro-insulin. Now, during the synthesis of insulin, what happened? That gene of the insulin is present on chromosome number 11. When that gene is stimulated, right, that produces messenger RNA, and mature messenger RNA, when it translates, that mature messenger RNA make a very long peptide which is called pro-insulin. Now, this long peptide chain, it enters into endoplasmic reticulum. When it enters into endoplasmic reticulum, this part of the chain is broken away. This piece of chain is broken away, and this is called signal chain, which help the pro-insulin to enter into endoplasmic reticulum of the cell. Now, what is left over here? The remaining chain is called pro-insulin. Now, pro-insulin has entered into endoplasmic reticulum. There it undergoes some modifications, and eventually this molecule, what is this? Pro-insulin molecule, right? Pre has been removed. Now, pro-insulin molecule will reach to Golgi complex. In the Golgi complex, what will happen? Another cut will be done. One thing which I want to say, that these two parts are connected with disulfide bonds like this. Now, when this molecule, what is this? Pro-insulin, right? This pro-insulin through the endoplasmic reticulum, while it is moving, right, eventually it reaches Golgi apparatus. In the Golgi apparatus, another cut is done. Another cutting is done. And due to that cutting, this remaining molecule, yes, consists of only two chains. One chain is this, other chain is this, and connecting chain is removed. This connecting part is removed. This connecting part is called C-peptide. What is it called? C-peptide. So C-peptide is removed, and this remaining molecule is called insulin. So what we can say, that in the end, in the Golgi apparatus, when secretory vesicles will be released into cytosol, these secretory vesicle will have 5 to 10% pro-insulin, and about 90% it will have insulin. So what is insulin? Again, suppose if this is the granule, right, insulin will have, let's suppose these two chains are insulin, alpha chain and beta chain. Some people call it A chain and B chain. They are held together by disulfide bond. Now this is what? Insulin molecule. And what is this? Pro-insulin molecule. And what is this? C-peptide. So these granules have mostly insulin, slight amount of pro-insulin, 5 to 10%, and but very important, the number of molecules of insulin and C-peptide are absolutely equal, right? Number of insulin molecules and C-peptide molecule is absolutely equal. They are absolutely equal. If there are 1 million insulin molecules, there should be 1 million C-peptide. either 1 billion, what is this? Insulin molecules, there should be 1 billion C-peptide. Now, with that, there is other things also here. For example, there is another substance called amylin. What is it? Amylin. Then another substance which is there, that is called, we have proinsulin, we have insulin, we have C-peptide and amylin. Now what happens? When lot of calcium comes, right, calcium acts on certain molecular motors. As I said, that glucose comes, right, glucose comes through the GLUT2. In the cell, glycolysis become more, Krebs cycle is activated, electron transport chain is activated, more ATP is produced. This ATP is going to block the potassium channels, and that will lead to hypopolarization. Hypopolarization, that membrane is less polarized, and that will lead to activation of voltage sensitive calcium channels. A lot of calcium will come in, and that calcium which stimulate certain molecules, we call them molecular motors, which will drag this secretory preformed insulin, pull it towards the plasma membrane of the cell, and eventually this will move and fuse it over here. And what will be released? Number one is, yes, most important substances. What is it? Insulin. Then small amount of proinsulin. Proinsulin you can forget because it's not important. Then with that, what is there? C-peptide, and then there is amylin, right? Now, I will talk about insulin and C-peptide later, just about amylin. Amylin has some functions like incretins. An easy way to remember amylin is that amylin has its function somewhat like incretins. For example, amylin can stimulate the beta cell and inhibit the glucagon secretion. Amylin can slow down the gastric motor activity. Amylin can act on the satiety center and lead to the feeling of satiety, that you are full, right? Now, I want to talk about C-peptide and insulin. When they are released in portal circulation. Where they are released? They are released in, now, what is happening here? Number one, insulin is going, right, in the portal circulation. Secondly, C-peptide is going, is that right? Then pro-insulin is going, which is not important, we can forget about that. And of course then amylin is going, amylin. With that, even incretins are going. You remember? Incretins. And here I would love to tell you, insulin, amylin and incretins, all of them can act on the hypothalamus and produce the feeling of satiety, that you don't want to eat more. What is this? This is defense against hyperglycemia. Once you are full, you don't want to stuff in more. You see it? Once you are full enough, right, and these substances are being released, especially some substances, amylin, incretins and even insulin, they have many functions, but one function is that they, through the blood, eventually they reach to the hypothalamus, and in the hypothalamus, ventromedial side of the hypothalamus has satiety center. So they stimulate the satiety center and you have the feeling of fullness. Is that right? Of course. Along with these substances, insulin, proinsulin, C-peptide, amylin and incretins, all of them are going through portal circulation. They are going, their portal circulation is also taking with it nutrients. So what else is going here? Glucose is going on, amino acids is going on, yes, fatty acids are going on, right? All of them are going to the liver. Now, liver is the next organ which will be exposed to the glucose and insulin. And here, insulin will work on liver cells, hepatocytes. Now remember, the tissue which are very insulin responsive, the tissues which have high concentration of insulin receptors. That is number 1, hepatocytes. They are insulin sensitive. Liver cells. Number 2, muscles, skeletal muscles and myocardial muscles. They are also insulin sensitive. And then adipose tissue, your fat tissue, adipocytes, fat cells, they also have insulin receptors. Rest of the body tissues have less insulin receptors. If someone asks what are the organs which are strongly influenced by the insulin, you will say number 1, liver, number 2 is skeletal muscle and cardiac muscle, and number 3 is your adipose tissue, right? Rest of the tissue have very less insulin receptors, right? Now, we come back. Now, first thing which we have to see it, that blood is passing through, entering into liver. This is now portal circulation and this is systemic circulation. We want glucose levels to be well controlled in systemic circulation, and liver is between the portal circulation and systemic circulation. Liver is very strategically placed between the portal circulation and systemic circulation. Let me show here. I will show you not only liver circulatory system, because that is very, very important to understand how liver handles the glucose and other nutrients in the presence of insulin in the fed state. Now, let's suppose that here is your liver, right? Rather, let me make it a little down and smaller. Let's suppose here is your liver. Here was your gastrointestinal system. Here was your pancreas and their spleen and all blood. This was which blood? Portal blood. This was all together coming to where? Liver. Through the portal vein and bringing insulin, proinsulin, C-peptides, amylin and incretins, and also bringing here nutrients like glucose, amino acids, fatty acids and other nutrients, right? Now, actually this portal vein will divide into smaller veins, right? And those smaller veins will divide into very small channels which are called sinusoids, and then sinusoids, listen, these sinusoids, hepatic sinusoids will unite together and make a central vein, and eventually central vein will go out as hepatic vein. Hepatic vein. So inflow is what? Portal vein. Portal vein, and also inflow is hepatic artery. Hepatic artery. Hepatic artery. So blood which is brought to the liver, that is number 1, hepatic artery from systemic circulation, and blood is also brought most importantly from the portal system, which drain the gastrointestinal system mainly, and pancreas and associated glands, and also drains the spleen. Now, actually this blood from the hepatic artery will join the sinusoids, will join the sinusoidal blood, and this will be also become mixed, and eventually all this will exit out. In between these sinusoids there are cells which we call them, what are these? Hepatocytes. These are hepatocytes. It's a very simple diagram. This is hepatocytes. So what is happening? Blood is passing by the hepatocytes. Blood is passing by the hepatocytes while hepatocytes membrane are working on the blood, for example taking up the nutrients or releasing the nutrients. Now let me make it more technical. This arrangement is not exactly like this, right? Let me make it more technical. Up to now you only understand that portal vein is bringing blood with nutrients and there is, what is this? Hepatic artery also bringing, and their blood mix into very small sinusoids, and outflow is hepatic vein, and hepatic vein will eventually drain into inferior vena cava. This will drain into inferior vena cava. There are 3 hepatic veins, and eventually they will fuse together and they will drain into inferior vena cava, which is the part of systemic circulation. What I am trying to put in your mind is that whatever you are absorbing from the GIT and whatever hormones are coming from the pancreas and from the gastrointestinal system, first they have to pass through liver, they have to be exposed to the hepatocytes, or they have to be modified by the hepatocytes. Once hepatocytes have acted upon the blood coming from the hepatic artery and blood and its content coming from the portal vein, only then blood will escape to the systemic circulation. It means if lot of glucose is coming and if it directly go to systemic circulation without passing through the liver, if blood goes directly to systemic circulation, hyperglycemia will occur. The function of the liver is very beautiful. What is the function of the liver? Liver has a nature like a mother. When more glucose is coming from the gastrointestinal system, liver will absorb the glucose and store the glucose in different forms. I will explain every step, but for a while you just trust me. When you have eaten a lot of food and lot of nutrients are coming, let's suppose glucose is coming from the portal circulation to the liver, then hepatocytes will take up those nutrients. Specifically, I am talking about glucose. Hepatocytes will take up that glucose, use that glucose for energy, use that glucose and make it in storage form like glycogen, and if still you have extra glucose, then they will convert extra glucose into triglycerides. It means hepatocytes have a capacity to convert extra glucose carbons into what? Into fatty acid chains and glycerol, convert into fatty acids, and those fatty acids attached with glycerol convert into triglycerides, and then triglycerides are released into blood as a part of VLDL, very low density lipoprotein. Let me repeat it, it's worth it. Thanks God that liver is present between the GIT and the systemic circulation, because liver acts as a buffer, especially for glucose. When a lot of glucose is coming to liver and passing through the system, right, what will happen? That these cells will take up the glucose and utilize the glucose for energy and convert the glucose into storage form for the future, that when you are fasting, when you don't have food, I will show you that same cells will stop taking up the glucose and start producing the glucose from the storage site and release the glucose to the blood, so that hypoglycemia does not occur. Again, in fed state, liver is like a mother. If there's too much nutrients in the home, she stores it. But when there are no nutrients, mother will release those stored nutrients and give it to the children. So liver is very selfless. It is not selfish organ. It will take the glucose in fed state in good times, store it for the bad times, and in bad times liver will convert into glucose producing organ. But because we are talking about this man who is going in jungle with full stomach, so at this time liver is in a situation we call it fed state, and in fed state it means liver is supposed to take up the glucose. Let me show you a little detail about the liver lobules. Liver lobules are like this, right? I will make one large here, right? Now what really happens, that portal vein comes over here and it will, from it branches will go at every angle. What is this? These branches which are going at the outer part of this liver lobule, these branches are called hepatic venules. What are these called? Hepatic venules. So hepatic venules are present all over here. right? And of course they are connected with the, sorry, they are portal venules. These are portal venules. Please correct it. These are portal venules. Now, from the portal venules, now I will bring one out. At every corner, what is there? Portal venule, right? Now, what will happen? At every corner, also a branch will come from hepatic artery. It will also divide into many branches. And at every corner, there will be one branch of hepatic arterioles. Hepatic arterioles, right? This is coming from portal circulation, right? And this is coming from hepatic circulation. Is that right? Now, in the center of this liver lobule, there is a, we call it central vein. What we call it? Central vein. And both of these there are special channels, vascular channels, which are highly permeable, and blood is flowing into these channels from periphery to the center. Blood is flowing from the periphery to the center. And portal venous blood and arteriolar oxygenated blood, both of them mix into these special channels which are called sinusoids. What are these called? Sinusoids. So what we can say that sinusoids are taking the blood from peripheral part of the each lobule, and these sinusoids are taking the blood to the central vein. In between the sinusoids, now listen carefully, in between the sinusoids there are, what are these cells? Hepatocytes. These are hepatocytes. Now, glucose has arrived. What is happening? Glucose has arrived through the portal vein, and glucose will go into where? Into sinusoids, because this is drained into sinusoid, also this also. Now, this glucose will drain into sinusoid. When it is passing through the sinusoids, these hepatocytes will start working on this glucose. They will start taking up the glucose. And remember, in between these cells, on the back sides of these cells, front of these cells is towards the sinusoids, right? Front of these cells are towards the sinusoids. On the back, there is a drainage system which is basically bile canaliculi. What is it? Bile canaliculus. So in this way, there is this thing which is called hepatic portal triad. What is it called? Portal triad. So in the peripheral part of the lobules, there are portal triad with the portal vein and hepatic arteriole and bile canaliculus. Is that right? And now listen carefully. Blood is moving from periphery to the center, and bile is moving from center to the periphery. But right now we are concentrate only on blood flow. Now this blood flow, which has come to all these organs, lobules, right? All of that blood is moving to, what is this? What is this? Central vein. And what are these? Sinusoids. And from the peripheral of the lobule, sinusoids are taking blood, portal blood, also hepatic arteriolar blood, through the sinusoids towards the central vein. And then these central veins, they will unite together and they will make hepatic vein, and that will go to inferior vena cava. So this is what is happening, that blood from the gastrointestinal system and from the spleen comes into portal vein, and blood does not go directly into systemic circulation. It has to be exposed to the hepatocyte first, so that hepatocyte work on the portal blood, and glucose and nutrients and insulin, all these things are coming through the portal blood, right? Now, what we see here, let's suppose we are talking about this special cell. It is receiving insulin, it has insulin receptor, and under the influence of insulin receptor and insulin stimulation, its metabolic pathways and enzymatic system will be altered under the influence of insulin, altered in such a way that more glucose will be used, more glucose will be stored. Is that right? And only then, and very, very important thing, when the blood which is leaving, the concentration of glucose will be less. So blood which is incoming blood to the liver has a high concentration of glucose, but because in the fed state, as hepatocytes are taking up the glucose and using the glucose and storing the glucose, so the blood which is leaving the liver, right, that blood is having less glucose, right. So it means here the liver behaved as a defense against hyperglycemia. Liver, by absorbing and retaining the glucose and processing the glucose, right, liver did not allow hyperglycemia in systemic circulation. Is that right? So liver acted as a defense against hyperglycemia in the fed state. Am I clear to everyone? Any question up to this? Now, not only if you take portal blood sample, glucose level will be higher. If you take hepatic vein glucose level, then what will happen? Blood glucose level will be down. But not only blood glucose, there is another thing, that is about insulin. About 40 to 50% of glucose is taken up by the liver, right? Whatever is coming through the portal system. And about 50% of the insulin, listen very carefully, about 50% of the insulin which was coming from this portal system, that is also, yes, also catabolized in the liver. About half of the insulin is catabolized in the liver because liver cells have an enzyme called insulinase. Insulinase enzyme will catabolize about half of the insulin. It means that if you check the level of insulin in portal vein, it will be high. But if you check the level of insulin in systemic vein, it will be low. So when we take venous sample, and if we try to check the insulin level in venous sample, it is not truly representative of what insulin was originally produced, because lot of insulin has been lost in the liver in the first pass effect. Am I clear? Here there is one very beautiful point. You remember that with insulin there was release of the C-peptide. In the beginning I told you that when insulin is released, right, with that connecting, I think you shouldn't forget, this was our insulin, and what was this? C-peptide. I told that from the beta cells of pancreas, C-peptide molecules and what is this insulin molecule, they are released in equimolar amount. They are released in equal number. But, so it means in portal blood, if you check C-peptide, and in portal blood, if you check the insulin, they will be the same concentrations. But the blood which is going out of the liver, right, insulin level will be low, as almost half of the insulin is catabolized within the liver, right, while it was doing its work, right. The point which is beautiful is hepatocytes do not destroy the C-peptide. Hepatocytes do not destroy the C-peptide. It means that if 100 insulin molecules were released, then 100, what is this? C-peptide were there. Only 50 insulin molecules will reach here. 50 insulin molecules will reach here. But how many molecules of C-peptide will reach there? 100 C-peptide. So which is more better representative of endogenous release of insulin? Insulin in our systemic circulation or C-peptides in our systemic circulation? C-peptide, because in the laboratory test we are taking sample from systemic circulation. In systemic circulation, when blood reaches, right, in systemic circulation, when blood reaches, it has already passed through the liver. Liver has destroyed almost half of the insulin. So insulin present in systemic circulation is really not very accurate representative of beta cell function. But C-peptide, because it is not destroyed in, what is this? Hepatocytes. So from the portal circulation, it passes through the liver, and without any destruction, it goes to systemic circulation, and you can catch it through the venous sample. And in systemic circulation, the amount of C-peptide, concentration of C-peptide is the true representative of release of what? Insulin from the beta cell. Am I clear? Next point about C. That is why if someone has hypoglycemia, or in someone, if you give exogenous insulin, when you give exogenous insulin injection, right, subcutaneous or intravenous, whatever form, if you give exogenous insulin, you don't give with it C-peptide. So if in the blood insulin is high and C-peptide is low, it means insulin is from exogenous source. Insulin is from exogenous source, right? But if in the blood C-peptide levels are very high, it means insulin which is present over there, that is from endogenous source. So actually levels of C-peptide more accurately represent the insulin production by the endogenous sources, that is beta cell. If I have a lot of insulin and no C-peptide, it means I'm injected with insulin. Is that right? Yes. But if I have more C-peptide and less insulin, it means that it is coming from inside. Yes. Am I clear? Now onward, what we will do, that we will take one cell, one hepatocyte. What we will do? We will work on every organ. Now I'm going to work on liver, not eye. Insulin is going to work on the liver. Now we'll take one big hepatocyte, right, and we will see how insulin acts on the hepatocytes, and under the influence of insulin, what changes come into different metabolic pathways in the hepatocytes, and we will see that whatever glucose is entering under the influence of insulin and fed state, that glucose passes through which metabolic pathways, right, depending upon the alteration in enzymes. So now onward, after the break, now we are going to take a break. After the break, we will concentrate mainly on a hepatocyte, glucose and insulin working in fed state, and what is happening within the hepatocyte to use the glucose and to convert the glucose into glycogen, and very special thing, how hepatocyte convert excessive glucose into fat, right? That we'll discuss after the break.